Okay, yes. So I've been asked to talk about ethical implications, so I suspect that I'm going to leave everybody with more questions than answers here, because they're ethical issues. Usually there is no right or wrong, they're just cause for thought, really. So just a bit of background. I am from the University of Birmingham, and I run the Human Biomaterials Resource Centre. For an academic institution, we are incredibly lucky to have a fairly new and shiny tissue bank. The floor plan indicates obviously we've got nice storage areas, cryostore, -80, big processing lab, all the swish stuff that you would expect a nice biobank to have these days. And we are a fairly typical research tissue bank. We have an HTA license, we have the generic ethical approval, and we spend most of our time trying to keep the rather demanding local research community happy. We have, in the last few years, like several people in this room, I think, been involved in a number of stratified medicine initiatives. Am I allowed to say that word? The first one for us was the CRUK Stratified Medicine Phase 1. Probably lots of you are familiar with this. I know some of you have been involved, where over a period of about 15 months, we were involved in the recruitment, consent and collection of both a fixed tumour tissue sample and a blood sample from getting on for 500 patients in the solid cancers listed there. And those samples all went to our local technology hub for a genetic analysis, which was at that time just based on a mutational pattern specific for the individual tumour type. So that was our first delve into stratified medicine. Of course, the clinical data set that goes with it was challenging, and that meant that there was a lot of informatics input at that time to try and get CRUK what they wanted. So then we morphed into CRUK Stratified Medicine Phase 2, which we've been in for about the last 18 months or so. And this is the collection. This is now focused just on late stage lung cancer. Again, it's a genetic analysis. We're actually doing the consenting, recruiting of the patients. We're chasing a fixed tumour biopsy sample from these patients, often from hospitals not where they are now. So this is quite challenging to get diagnostic tissue biopsies from another hospital, and a blood sample, and they're going for genetic analysis on a bigger mutational platform. Now, I don't know how many of you in this room will be aware of the fact that the way this is set up now is that Stratified Medicine 2 is essentially now the pre-screen for the National Lung Matrix trial. And the National Lung Matrix trial is a trial whereby there are actually several arms to it, with targeted therapies, specific targeted therapies in each arm. And based on the genetic results that come back, the patient would be stratified down to one of those arms. And I just want to spend a few minutes talking about it, because this trial has only just begun. We've just started recruiting patients in Birmingham, and it's the first time that up front I've seen how the patient is absolutely desperate to get that result and be stratified and be given an opportunity to get a drug. To the extent that they are phoning us, chasing the blocks, they are phoning, getting phone calls, trying to get phone numbers for pathology labs, and trying to really speed it up. So I've been staggered by that, and it actually makes the stratified medicine thing seem fairly real. First ethical issue, well actually second, the first ethical issue is the fact that these patients are presenting late. They are unresectable, so we have to chase their biopsy, which means we are consenting them at a time when possibly they haven't had their diagnosis. And it just highlights that in all these areas, when you consent patients and the pathways they're going through, actually you have to think about it, and it's different in different disease settings. Secondly, currently, and this is awful, is that currently 50% of those tissue DNA samples are failing QC, and they are not getting a result back. So the patients are being consented, given this hope for the trial, and then actually we don't get a result back, and so they are then offered the opportunity to be re-biopsied, which is not that pleasant in that setting. So we are still not, we still have, I think, issues with what we're trying to do with fixed tissue blocks, which I think in time will actually become much better, but there's a bit of work to be done because that's actually very disappointing. Okay, so then we moved on to the gel pilot, Genomics England pilot, and again within a very short space of time we consented and delivered a sample set on, I think it was nearly 400 patients. And in this case, this was the warm-up for the 100,000 project. In this case, actually, those samples went away, the DNA samples went away for whole genome sequencing, and the results were not fed back. So now then, Birmingham is involved in the West Midlands Genomics Medicine Centre. Is anybody else involved or working in it? I feel your pain trying to implement at the moment. And so the HBRC is actually what they call the central collection point, so that all the samples that are being collected will be coming back and going out from us. And we will also be processing all the omics samples, which I suspect the list might have come from UK Biobank, because it looks remarkably similar. Thanks for that. So we'll be doing that bit. And obviously, as you know, this is the government project, or the DOH project, which is going to be whole genome sequencing, 100,000 patients in Birmingham. I think possibly we'll be doing about 17,000 of them if we reach our trajectories over the next 3 years in both the cancer and rare disease setting. So we've got a bit of experience. So why are biobanks the best place for these sorts of projects? Well, clearly, as Tim pointed out, this is down to infrastructure really. It's really the place where there's going to be the facility, the infrastructure to actually do this to a high standard appropriately. Okay, I was going to define stratified medicine, but Tim did it, just because I actually think sometimes it's a bit of a term that people think slightly different things about. So basically matching therapies to specific patient groups using clinical biomarkers and obviously other characteristics. Interestingly, there's actually not much currently that is used in this way, in a targeted way. Probably the most classical example, the classic example, is Herceptin, which is obviously offered to about 15 to 30% of breast cancers where there's an overexpression of HER2. But there aren't actually that many other examples in routine clinical practice at the moment. So obviously we want to adopt this. We've all historically had a fairly not ad hoc approach, but we no longer want to do this one size fits all, which means that you're just treating a whole patient group with exactly the same drug. Some of them will have terrible side effects, some of them will do well, some of them won't. So the whole point is to actually make that a lot easier. Classically, well, actually in theory, you can stratify using any biomarker that you can validate as being appropriate to do it. So I think most people in this room would accept that the way we're going to make the most progress in the short term is to use the genomics medicine approach rather than the others at the moment. So obviously this talk is centred mainly on genetic analysis, and of course that's where predominantly the ethical issues are. So I'm going to start talking about the ethical issues. This is not going to take long because there isn't actually, these are food for thought and points for discussion rather than me lecturing about what's right and wrong. The first issue I think about stratified medicine, particularly in the genomic medicine, is obtaining what we all like to think of as an informed consent. So I wonder quite a lot about how much information, when you're consenting a patient to a genomic medicine project, they actually understand. Because certainly some of them don't even know what the word gene means. They certainly don't know what the word genome means. And I just wonder, I wonder whether you are aware of the fact that the 100,000 K, or 100,000, no, that's too many noughts, 100,000 genome project, the information sheets and the consent forms are actually several pages long, and in my opinion, not in that user-friendly language. So I do wonder that in our efforts to actually obtain an informed consent, we're actually obtaining an uninformed one these days, because actually the participants don't understand what the information that they're being given to read. Incidental findings, do they really understand what that means and what the impact an incidental finding will have to them and their family? Before going into disease things, what about if they find out that their parents aren't who they thought they were? Just a really basic thing, paternity issues. In the consent forms for the 100,000 project, they have to opt in to having results fed back about their disease condition, but they have an option of being fed back findings, what's called secondary findings or additional findings relating to diseases that we might be able to do something about. And also they have an option of opting in to be fed back about their carrier status in some diseases. Do they really understand that complex information that they're being asked to choose? Do they understand that access to health records means that actually now, at any time in the future, people can go and look at their notes and look at their personal information? I don't know. Okay. Obvious second issue is protecting their privacy and autonomy. Should the patient have to share their data as a condition of taking part? So they're having their profiling done because they want to know whether there's any indicators for them. But does that mean necessarily that it's okay to make that data go into a pool to actually inform other people's profiling and diagnosis? I don't know. Who should the information be shared with? I think that we would all agree that the information is very valuable for research-based population and to actually further the stratified medicine initiatives. But we all get a bit scared about the mention of insurance companies and future employees. Lots of people think, because obviously what we do, our bog standard way of coping with this is to anonymise. Lots of people think that actually you can't anonymise genetic data. A whole genome sequence is actually a huge personal identifier. And what about the impact of findings on family members? Have we got a right to feed back findings to a participant which will then directly impact on their family? Have we got a consent issue then with the family, with the relations? I think we probably have, and 100K project has addressed this in that they are recruiting duos and trios, and they too are obviously consenting to feedback of findings. But the whole thing is tricky. Treatment options and decisions. This is something that's wasn't so obvious to me till I started to think about it. And really we're talking about at some point in the future now, when we have done our stratifications, we have got drugs that we know are better suited to certain subgroups of patients. And our patients are and we have a validated screen for that. So what happens then when you have to return the results of that data to a patient? And actually what you've got to tell the patient is that there is no effective treatment, or that their options are very much reduced. Or even worse, if the actual drug that's available or the drug that's going to work is not actually available on the NHS. So this sort of information will have a big psychological impact on the way you feed back information to patients and decide. What if the clinician and the patient disagree? So the clinician is saying, no point in giving you that drug because the probability of you responding is not going to be very high, and the patient is actually desperate to have the drug because it's his only option. Who makes that decision as to whether or not they actually are prescribed the drug? And what happens to a patient who actually doesn't want to do the screen, doesn't want to do whole genome sequencing because they don't want to undo that can of worms and all the ramifications of that? But the drug is only licensed for patients who with a certain genetic profile. How have you got a right to actually say, well, no, we can't prescribe you that drug? All of these things possibly later on down the line will become difficult to handle. Finally, resources. So at the current state and time, these initiatives are hugely expensive. They are costing the NHS a lot of, well, the 100k genome project will be costing the NHS a lot of money. They're expensive, you know, and they're a huge, or they will be quite a burden on the healthcare systems. There's an increased education and training need, genetic counselling, more time needs to be spent with the patients in the clinic for the consent process, which takes 25 minutes to do it properly. Pharma are not likely to be happy to fund much of it because they want to develop drugs that are going to be useful for huge populations and not smaller ones. And we've talked a bit about the informatic challenges, and they are massive. Trying to link up these huge bits of genetic information with a clinical record in a way that makes it meaningful and easy for a clinician to actually decide treatment is no mean feat, and it's ongoing. In fact, in the West Midlands, one of the legacies that we think for us that will come out of the 100,000 project is the ability actually to join up informatics across the region, and that's one of the main drivers for us wanting to be part of this. So there are some people who would think, well, all of this being put into this, when there are some countries in the world that haven't even got sanitation or basic immunisation or vaccination programmes, or even access to the very common medicines, food for thought. So in conclusion then, I think stratified medicine, particularly in genomic medicine, is obviously currently a rapidly developing field. It does definitely have the potential to cut the cost of ineffective drug treatment within the NHS, and to make the patient or make patients' lives a bit nicer by reducing adverse reactions. But it clearly presents a lot of challenges at the moment, not only the ethical ones, but basically the complexity of trying to develop and assess a medicine and a genetic test at the same time. Thank you very much.